This is podcast number 37. We're going to start this podcast off with a couple of HIP questions that go back a bit. And one of the last specialized cell types we'll talk about, the last one being HeLa cells, is muscle. And your book doesn't deal a lot with muscle. So most of these figures are coming from not your book, but other sources. So there's a general phenomenon in muscle cells, which are a type of excitable cell, like neurons are, where there's something called calcium-induced calcium release. So that release of calcium from another cell, usually a neuron, will cause calcium to be released from inside the muscle cell, and that's the calcium-induced calcium release. Inside a muscle cell, there's a specialized version of the endoplasmic reticulum that is called the sarcoplasmic reticulum. Sarco meaning muscle. And in general, calcium is high outside the cell and low inside the cell. However, the endoplasmic reticulum in any old cell will store calcium, and especially in muscle cells. So this is thought of as a calcium store, a storage place for calcium, and it can be released from the sarcoplasmic reticulum to the cytoplasm in the cell. Sometimes they just refer to the calcium, internal calcium, as calcium stores, but they're talking, researchers, when they're talking about this, they're talking about calcium that is stored inside the endoplasmic or sarcoplasmic reticulum. So again, calcium is going to play a big role in how muscles contract, and calcium is first released outside the cell, causing calcium to be released inside the cell. A lot of this was figured out in this little beastie here. This is an Opsanus tau, the oyster toadfish. Here is its eyes, its mouth. It's a bottom dweller, pretty voracious eater, and they have a specialized band of muscle around their swim bladder. And that band around its swim bladder, when it vibrates really, really fast. And it sounds just like you're pulling a thumb across the surface of a balloon. Swim bladder is like a balloon, and when you do that, you hear that sort of humming sort of sound. And I can play a little video here. Let's see. This is a different species of toadfish, but you could. All right, go back here so you can hear that again. So that's the sound that the toadfish make when they are defending a territory or when they are breeding. And it's loud enough that a bunch of toadfish can produce enough sound that you can hear it above the water. Usually where these guys live, this species here is like East Coast, Cape Cod, that area. There's usually a story about the bays being haunted because they hear these toadfish, excuse me, but they aren't ghost toadfish. They're just toadfish looking for love. So this muscle that surrounds their swim bladder contracts very, very fast. It's the fastest contracting vertebrate muscle, up to 200 cycles of contraction and relaxation in a second. That's what Hertz is. You might think of other things that you would think would be faster, like a rattlesnake shaking its tail or a hummingbird flying. They're actually not as fast as this swim bladder muscle. And there are faster contraction muscles, such as in a honeybee, in their flight muscle. But in honeybees, they cheat a little bit because they have the pulling on the the elytra, the external skeleton of the honeybee to pull the wings up when the muscle contracts, and then the physical properties of that bee exoskeleton causes it to pop back into its original shape, which is part of the relaxation cycle, and that's how they can make it faster. So they only have to contract for half of that cycle, and then the physical properties of their exoskeleton causes aids the relaxation part of the cycle. So toadfish swim bladder contracts and relaxes really fast, and these researchers figured that was a great place to figure out this excitation contraction coupling and the calcium induced, calcium released, because they figured that whatever they're doing, they're going to be doing a lot of it because it's so fast. This person was my postdoc advisor. This person was her postdoc advisor, and this person, the Armstrong and Franzini Armstrong, was the person who discovered the ball peptide in the ion channels that we just talked about in neurons, the inactivating thing that plugs up the sodium channel after it opens so that it's inactivated for a short amount of time, allowing the signal to go only in one direction. That was Clay Armstrong. So lots of muscle brain power here. Okay, so what did they figure out? Well, first you got to review some of the anatomy of a muscle cell. When a muscle physiologist talks about muscle fiber, they are talking about a single muscle cell. And in adult vertebrates, that single muscle cell is generated in development from fusing together multiple muscle cells, so that single muscle cell will have multiple nuclei, and it can be a very large cell, up to 1 millimeter in cross-sectional area, which is really big for a cell. In those muscle fiber cells, and the muscle fiber would be cells would be collected into a bundle, which eventually becomes a muscle with connective tissue, like a biceps brachii or something like that, or a deltoid muscle or trapezius muscle. But if we're talking about a muscle cell, that's a single cell with one membrane around it. The membrane around it, the muscle cell, is given a special name, sarcolemma. So instead of the plasma membrane or cell membrane, like we have been talking about for other cells, for muscle, it's called a sarcolemma. And instead of endoplasmic reticulum, we say it's sarcoplasmic reticulum. The muscle cell interior is dominated by bundles of actin and myosin that are going to contract against each other, and those bundles are called myofibrils. And in addition, this sarcolemma, the muscle fiber cell membrane, has invaginations that poke down into the belly of the muscle. These are called T-tubules or transverse tubules. So the transverse tubule is connected to the sarcolemma, and you can think about it like if you had saran wrap, cling wrap over some jello. The saran wrap would be the muscle cell membrane, the sarcolemma, and the jello would be the inside of the muscle cell, all of this stuff in here. If you poked your finger into the saran wrap, not hard enough that you're going to break the saran wrap, but just gently enough so that your finger pokes into the jello without breaking the saran wrap, that invagination, that hole that you produce, covered up your finger now in saran wrap with jello surrounding it, that hole going into the belly of the jello or the belly of the muscle cell, that's the T-tubule. So anything that is happening to the cell membrane here is also happening to the T-tubule because the T-tubule is part of the cell membrane. And that's key to understanding how this works. Okay, here's another diagram that you might find more useful. So if we take a section out of a muscle, there's going to be several muscle cells wrapped up in connective tissue. If we look at just one muscle cell, one myofiber, there's going to be several myofibrils. Within that, myofibrils are the actin and myosin bundles within the muscle cell. That muscle cell is going to be surrounded by a plasma membrane, which is called the sarcolemma. That plasma membrane is going to have deep invaginations that reach the belly of that muscle. Those are called T-tubules or transverse tubules. There will be an extensive network of endoplasmic reticulum to store calcium. In the case of muscle, that's called sarcoplasmic reticulum instead of endoplasmic reticulum. Lots of mitochondria, etc. Okay. This is the model that Bloch and Franzini Armstrong came up with for how calcium being released on the outside of the cell causes calcium to be released on the inside of the cell. And this is only figured out relatively recently, in the late 80s is when this was, 1980s is when this was figured out. And you would think for something as highly studied as muscle, we would have a better idea for how an action potential arriving on the outside of the muscle cell causes muscles to contract, but we honestly did not. The part between the action potential and the muscle contracting was missing. So here's how it works. The T-tubule is part of the sarcoplasmic reticulum, okay, which is part of the outside cell membrane, and is just dipping down into the belly of the muscle cell, okay? And remember, these muscle cells are really big. So if you are relying on diffusion to have things reach the middle of that muscle cell, you'd be waiting a long time because it takes a long time for something to diffuse across something as wide as a millimeter. So an action potential arrives at the outside of the muscle cell, and it causes a depolarization of this muscle cell membrane. Remember, we just talked about this when we talked about neurons. Is the charge on the inside relative to the outside at resting is negative, about minus 60. And when an action potential arrives, it can be temporarily positive and then go back to negative. That's depolarizing the membrane. Because the whole membrane is connected, the whole membrane gets depolarized at the same time. And that depolarization is sensed by a protein that is a voltage-gated channel that's in the membrane of the T-tubule. Okay? We'll talk more about the details of that protein in a second. But it's a voltage-gated protein. That voltage-gated protein is physically connected to another voltage-gated protein called the ryanodine receptor that is now connected to the sarcoplasmic reticulum, and that's inside the cell. So you have one voltage-gated protein that is changing shape, and it is connected to a voltage-gated protein on the inside of the cell. Now I said it's a voltage-gated protein, and it is, but it's also a ligand-gated protein. It reacts to calcium and not only calcium, and I'll talk about that in a second. So when this first protein that is voltage gated and connected to the T tubule opens up as a result of sensing a change in voltage, a little bit of calcium comes in from the outside of the cell because calcium is high on the outside of the cell. A little bit comes in. That little bit of calcium, in addition to these two being physically connected, causes this channel on the inside of the cell that is part of the sarcoplasmic reticulum to also open up. And the sarcoplasmic reticulum is full of calcium. Calcium floods out of the cell. Floods out into the cell. Floods into the cytoplasm of the cell from the sarcoplasmic reticulum. Calcium is going to cause contraction to happen, and we'll get more into how that happens in the next podcast. But then for relaxation to happen, you've got to get rid of this calcium that is in the cytoplasm and put it back in the sarcoplasmic reticulum. That happens by a pump that uses ATP to move calcium against its concentration gradient from relatively low concentration in the cytoplasm to relatively high concentration in the sarcoplasmic reticulum, and it pumps that back in, and that will cause relaxation. So, action potential arrives at the muscle cell. You decide you're going to contract that muscle. It depolarizes the muscle cell membrane, the sarcolemma. There's a membrane protein bound into that membrane in the T tubules, and because the whole membrane is depolarized, it senses that change. It's a voltage-gated channel, and this is physically connected to a calcium release channel in the sarcoplasmic reticulum called the ryanodine receptor. A little bit of calcium comes in, causes a bunch of calcium to come out of the ryanodine receptor, out of the sarcoplasmic reticulum. Calcium causes contraction. We're going to cover that in the next podcast. And then in order to get relaxation, you need your sarcoplasmic reticulum to be activated, which pumps calcium out of the cytoplasm back into the sarcoplasmic reticulum with the action of ATP. Here is another diagram to show you how these things are arranged. Sarcoplasmic reticulum and the T tubule are all connected. Okay, they're all one part of one membrane. So if this thing in yellow is depolarized, then this thing in blue, which is the the voltage-gated channel that is part of the membrane, it's called the dihydropyridine receptor. That dihydropyridine receptor is going to change shape as a result of the voltage change in the sarcolemma. Because it's changing shape and because it's physically connected to the ryanodine receptor, which is the calcium release channel in the sarcoplasmic reticulum, that changes shape also. So this blue heart changes shape, and this red house changes shape as a result of that, and that releases calcium out of the sarcoplasmic reticulum. And then in order to get it back in, you use the calcium ATPase or SERCA to pump it back in. So this thing here, this arrangement here is called a triad. So you have a T tubule plus these connecting proteins plus sarcoplasmic reticulum. And you see triads a lot in voluntary skeletal muscle, like in that toadfish swim bladder, and that's why they were able to do this work in that creature, because it had a lot of this thing to look at. In heart tissue, in you and I, and in most vertebrates, these two aren't physically connected. So dihydropyridine and ryanodine aren't physically connected in heart tissue. And then that's where it's more calcium-induced calcium release, because the dihydropyridine receptor will open because of the voltage change, and it's voltage gated, and a little bit of calcium will leak out, and then it'll diffuse across a short distance and bind to the renin receptor, which cause a lot of calcium to come out. So that's the calcium-induced calcium release part. But in skeletal muscle, they're connected to each other. So you have one changing shape causes the other one to change shape. So how did these proteins get their names? Because they don't seem like intuitive names. They're named for drugs that bind to these things. So dihydropyridine is a drug that binds to that voltage-gated channel in muscle. That's part of the T-tubule. And ryanodine is a drug that binds to the calcium release channel in sarcoplasmic reticulum. And these drugs come from plants, and plants use them as defense. And they're super potent. So I worked on with ryanodine when I was a postdoc, and to get this little itty bitty 1-inch high bottle, it comes in a Russian doll nest of other boxes when you order it from a chemical company, and that the box that you get coming from FedEx is actually about 3 feet high and about a foot across, and you have to open about 3 different boxes that are full of chemical absorbing material before you get to your ryanodine, because it's so poisonous. Because ryanodine binds to these sarcoplasmic reticulums channels right here and prevents calcium release when it does bind. And if you don't have the calcium release from your sarcoplasmic reticulum, can't contract your muscles, you die. So for example, your heart is a muscle that's very important that it keeps on contracting, and if you can't contract it, you die. Even having said that, and we were really careful whenever we were working with it, in the 1950s they used to use ryanodine as an insecticide. So you can see these old movies on YouTube where they're just fogging people in a pool with ryanodine to prevent mosquitoes from biting them. They don't do that anymore. Okay. And the ryanodine receptor, this thing that I have as cartoon as a little red house here, is absolutely huge as proteins go. So 4 subunits, each one is 6,000 amino acids long chain. So 24,000 amino acids in this protein. Like I said, it's the way it works is it kills animals by binding to the calcium channels, prevents calcium release, that prevents muscle contraction. Like I said, it was a common pesticide, but no longer used because it's so dangerous. And there's a really cool story about how a mutation in this protein was identified. So there's a mutation in pigs called malignant hyperthermia. And it's not just in pigs, it's also, you can see it in dogs and in people. And it's a condition that is characterized by uncontrolled heat production. So all anesthesiologists learn about malignant hyperthermia, and they know to monitor a patient's body temperature as they're going under anesthesia. And if their body temperature increases dramatically, they have to administer an antidote quickly called dantrolene to bring them out of anesthesia and to stop this uncontrolled heat production. So the uncontrolled heat production comes from muscle microcontractions that are happening all the time, happening, and whenever muscles contract, they're going to use a lot of ATP. And if you are breaking ATP faster than you can dump the waste heat that is made from breaking ATP, any kind of chemical reaction when you break a bond, you're going to create some waste heat. If you do that too fast, your body temperature can increase at a rate that you can't get rid of the heat fast enough, and you literally cook from the inside out. So this was unwittingly selected for in hog farmers. So in the 70s and 80s, there was people started getting more conscious about fat in their diet and looking for leaner cuts of meat, leaner cuts of bacon, for example. In a regular old pig will look like this. And there were spontaneous mutations in these pig breeding colonies that made pigs that look like this. And if you notice, they're just really cut sort of pigs, have very good muscle definition compared to this guy over here. And these were thought to be desirous because of they were very lean. They had very low body fat. However, when these animals were driven to slaughter, so they put them into transport vehicles, or they herd them into a barn for slaughter, they would get stressed out, and as they got stressed out, some would fall over and die, and they would open up the pig to try to figure out what was wrong, and steam would escape from the pig. They identified the mutation that led to this as being a mutation in the renin receptor, and it's only one amino acid out of 6,000 that causes this mutation. And here, and these are just the sequencing traces of DNA sequencing to show which amino acid was different in these pigs versus a wild type pig. So how do you make sense out of this? So in these pigs, under stress, they were dumping more calcium than usual. And in the background, even when they're not under stress, their renin receptors were open more than in this pig, in a wild type pig. So this wild type pig, the renin receptors stayed closed unless they wanted to contract that muscle. In this pig, their renin receptors were open most of the time. So it doesn't mean their muscles were contracting all the time, but they were getting these micro contractions. So a little bit of each cell in a muscle was contracting all the time. And that made them very muscular because it's like they were doing crunches 24/7. And to fuel that, those muscle contractions, they were burning through their fat reserves. And so that made them very lean. And then when put under extra stress, like being driven to slaughter, now their muscle was being activated even more, and that was more than the pig could handle in terms of the heat that was generated. For people and for dogs, this can be exacerbated when someone goes under anesthesia. So under anesthesia, what can happen is the membrane fluidity of the sarcoplasmic reticulum is affected by the anesthetic. That's how a lot of anesthetics work, is by messing with membrane fluidity, and by messing with membrane fluidity, you mess up synaptic transmission, and that's why you don't feel pain. But by changing the membrane fluidity in this membrane-bound protein, bound in the membrane of the sarcoplasmic reticulum, now it's open even more so than usual. So humans who have this mutation and dogs who have this mutation have the same sort of physiology in that they're very lean, and they have very defined muscles. But then when you mess with muscle or with sarcoplasmic reticulum membrane fluidity under anesthesia, now the channels are full on open and the muscles are contracting faster, are contracting at a rate and generating heat at a rate faster than can be eliminated by ways that bodies eliminate heat, such as sweat and respiration and dumping blood flow to the surface. And so the rate of muscle contraction and the rate of heat rise as a result of muscle contraction can be so fast that it can kill the human or the animal. So in dogs, for example, it can happen that a person will drop off their dog at the vet to have their teeth cleaned. Dog don't like to have their teeth cleaned, and so they knock them out in order to do it. And the dog dies because they have this malignant hyperthermia. In humans, it's super rare. I know a couple anesthesiologists, one a former student and one just a friend, and they say they have never seen it in their career, but that they learn about it and are ready in case they ever do see it, and they do monitor people's body temperature as they are going under. So just a bit of background about ryanine receptors and how we learn more about how they work by this unwitting selecting for it in pigs. They don't do that anymore. Another thing about ryanine receptors is that caffeine binds to ryanine receptors. So before I said it's both ligand and voltage gated. Voltage gated in the sense of it's connected to another voltage gated channel. So a change in membrane voltage will open up the ryanine receptor in the case of skeletal muscle. In the case of cardiac muscle, heart muscle, it's ligand gated in the sense of it's not physically connected to the dihydropyridine receptor, and so a little bit of calcium comes across, and that causes the ryanine receptor to open because it's sensitive to calcium, but it's also sensitive to caffeine. And this is why caffeine can give you the shakes. If you have too much caffeine, it can cause your ryanine receptors to be open more than usual and can make your hands shake or other muscles shake because you are having these micro contractions because your ryanine receptors are open more than usual. Caffeine is also a common ingredient in diet pills for the same reason. It binds to ryanine receptors, causes calcium to be released more often, causes you to pump that calcium back into your sarcoplasmic reticulum, and all that costs energy and therefore costs you to lose weight because you're using energy at a higher rate. It can also cause problems with your heart and cause other sorts of physiological problems, so it's not a good idea to megadose on caffeine. And you can acclimate to it. That I know that if I avoid caffeine for a while and then I go back to it, then I'm really sensitive to it. But if I am on a deadline and I'm using a lot of caffeine in what I'm drinking, then it takes more and more to have the same effect of making you alert.